Okay, let's learn about DNA replication, how organisms copy their genetic information, their chromosomes, their DNA molecules, so that cells can reproduce, so that organisms can grow. If we can't copy our chromosomes, we can't make new cells because the chromosomes contain the information that cells need to function, to do the work of a living cell. And so, how do we unwind this double helix, this double stranded, twisted rope ladder of DNA, and make an identical copy of this molecule so that there will be two copies, one that can be allocated to each of the new cells when cells divide. One of the things that's hugely important about this process of replication is the specificity between the different nucleotides. Those are the monomers, the building blocks of DNA. And remember, there are four different nucleotides: A, T, G, and C, adenine, thymine, guanine, and cytosine. And because of the structure of the nitrogenous bases, the different parts of the four nucleotides, because of the structure of those molecules, hydrogen bonds are able to form very specifically between adenine and thymine and between cytosine and guanine. When you match them up in any other way, the atoms don't align correctly and hydrogen bonding cannot happen. So, because of this AT and GC specificity, we say that the adenine is complementary to the thymine, the guanine is complementary to the cytosine. They match up. Because of that, DNA kind of almost sort of replicates itself. It needs a lot of help, but it at least gets things lined up almost by itself. All right, so a little background information first and a little big picture information. In prokaryotic cells, those are like bacteria, the DNA is not in a nucleus, it's in the cytoplasm. And most prokaryotic organisms, most bacteria, have just a single loop of DNA that is their chromosome. It's not really the same as a eukaryotic chromosome like in our cells, but it is the same stuff. It's DNA. It also has proteins that package the DNA like our chromosomes do, but they're different proteins, so it is different. And then most bacteria also have smaller loops of DNA that are called plasmids, and those plasmids often contain sort of extra genes that may be helpful for that bacterium. And plasmids are frequently exchanged between bacteria. So genetic information can be passed back and forth between bacteria. It's kind of a way for them to have sexual reproduction. They're exchanging DNA with other individuals. And when we talked about bacterial transformation in the last section, Griffith's experiments with the dead bacteria and the live harmless bacteria, this was what was going on. There were plasmids that were being transferred between the dead and the living bacteria. So here's what that looks like. You can pause the video for a second, take a look at this. But this is a typical sort of bacterial cell. A single loop of DNA as the chromosome. That loop is often isolated in one part of the cell that's called the nucleoid. And then there are often one or many plasmid DNA loops bouncing around the cytoplasm. In eukaryotic cells, chromosomes are as we described them in the last unit when we talked about mitosis. And a typical eukaryotic cell has lots more DNA because the cells tend to be more complex. The sort of instruction manual for how to run that cell is a bigger book. more chromosomes. Chromosome number varies greatly between organisms and animals. The smallest, simplest organisms have just a few chromosomes, including only 2 in certain species of roundworms. That's just 1 pair of chromosomes. And in other animals, the number can be much greater. There are certain hermit crabs that have 254 chromosomes, which is crazy. Are they giant, big, complex organisms? No. And if you're curious about why those hermit crabs might have so many, and why a simple, small fern plant like this adder's tongue fern that's pictured here, why it would have over 1,000 chromosomes. So here's an electron micrograph showing the chromosomes from one of these fern plants. Here's an electron micrograph showing the chromosomes from a human. And that fern's got a lot more than we do. Right, so eukaryotic chromosomes have our DNA. The molecules of DNA are what's important for the information that the chromosomes contain. And then there are also lots of proteins that are responsible for packaging the DNA, keeping it stable and safe while it's being used or not used. When we wrap a DNA molecule around some of the proteins called histones, we create this beads on a string structure that we saw in the last unit. Those are called nucleosomes. The nucleosomes can then coil up around each other, forming thicker and thicker chunks that we call chromatin. Chromatin can coil up and form supercoils. The supercoils can fold up on each other, and then we eventually wind up with one of these thick condensed chromosomes that we see during mitosis. So how does replication occur? How do we get 2 identical copies of a molecule of DNA, of a chromosome? Well, each strand of a double-stranded DNA molecule provides the information for building the other strand. We know that the A nucleotides, the adenine nucleotides, are only able to form hydrogen bonds with thymine nucleotides. Likewise, guanine only with cytosine. And so if we have a double-stranded DNA and we're able to separate those 2 strands, we ought to be able to look at one strand and know exactly what the sequence of the other one is. And that's because of this complementary nature of the nucleotides. A always bonds with T, G always hydrogen bonds with C across the double-stranded DNA. And so the way it works in general is that the organism has an enzyme, it's called helicase, and the helicase enzymes get into a double-stranded DNA and essentially unzip it. They separate those 2 strands, and now those single strands provide a template for building the opposite strand. Because if we see what the sequence is on one strand, we know which nucleotides would line up and bond with the other strand because of this specificity between A and T, G and C. Those places where the DNA molecules are unzipped are called replication forks. And in bacteria, which have relatively small chromosomes, they just start at one place and go all the way around the loop. In eukaryotic organisms, which have much larger DNA molecules, there are multiple replication forks. So the cell will start copying the DNA molecules at multiple places along that long chromosome, so it doesn't take so long to get the process done. So DNA replication is this process of copying the chromosomes, and it results in 2 identical sets of chromosomes so that each of the new cells that will be formed by mitosis would have an exact set of chromosomes to the other one. So, and an exact set of chromosomes compared to the parent cell that produced those two new cells. Here's what it looks like in a little bit more detail. We see the enzyme helicase here on the parental DNA, which is still zipped up when you go towards the left of the screen here. And then you see the darker blue is the parent strands of DNA. So those were the original strands on the original molecules. And after helicase unzips the molecule, a whole cohort of enzymes jumps on the molecule to help synthesize the new complementary strands to the two parent strands. And if we go out here a ways on the template strand, we see that there's now double-stranded DNA. And the same thing is happening on the strand up above. So what we have created is two identical strands of the DNA that we started with. You don't need to know any of these other enzymes or any of the details about this process. You don't need to know about primase or topoisomerases or DNA ligases or different DNA polymerases. You just need to know that helicase unzips the DNA, and then a second enzyme called DNA polymerase is responsible for building the new strands. And here's what that looks like in a little bit more detail with a little less clutter. And again, don't worry about the other enzymes. The ones that you need to know are DNA helicase and DNA polymerase. The DNA helicase is here unzipping the parent DNA, moving, in this case, from left to right. And DNA polymerase enzymes are on both of those original parent strands, building the new complementary strands. And here we see on the ends of each of those new strands, double-stranded DNA that would be identical to the original parent DNA that we started with. Oh, and the DNA polymerase, in addition to attaching the nucleotides one after another, and you can see here on the leading strand, here are some individual nucleotides that are lining up on that single-stranded parent DNA, and they would line up with those base pairing rules that we talked about, A with T, G with C. And they're all sort of lined up there, ready to go, and the polymerase enzyme comes along, makes sure the correct nucleotide is in place, and then attaches that free nucleotide to the growing strand of new DNA that's being built on the parent strand of DNA. Just another picture to give you another image. I like this one because it's showing the free nucleotides falling into place here. Again, those nucleotides are going to tend to only line up with their complementary partner, AT, GC. And before replication would have begun, in preparation for copying the chromosomes, the cell would have made a whole bunch of free nucleotides, a whole bunch of A's, T's, G's, and C's, and put them in the nucleus, ready to be used to build these new strands of DNA. And then this one gets right down to a molecular level, right down to an atomic level, and shows what happens when a new nucleotide is added to a growing strand. So the strand on the left side here, the pink one, is the new strand. The blue is the parent strand. After the DNA had been unzipped, we see that the A's and T's, G's and C's are matching up. And here on the parent strand, the next free nucleotide is a guanine. So the next free nucleotide that would match with that that would hydrogen bond with that would be a cytosine, a C. And so that happens spontaneously. This C would just be attracted to that G and line up in that spot. And then the enzyme DNA polymerase, moving down along the parent strand in this direction from top to bottom, would come upon this cytosine that has hydrogen bonded to the guanine. It checks that pairing and says, yep, C with G is correct. And then it catalyzes the covalent bond to attach the phosphate group of this nucleotide to the sugar of the last one on the strand. And that's how, one nucleotide at a time, the new strand of DNA is built.